Good morning, everyone. I am Kirandeep Kaur of class 12C. Today, I am going to give an explanation on a biological topic like operon. Lac operon. Lac operon generally is going to operate in the case of prokaryotes, and this works generally for the consumption of lactose. And lac operon is generally an inducible operon. Inducible operon means it only going to operate in the case of the presence of lactose. Here our lac here indicates lactose. So in the presence of lactose it's going to function, and in the absence of lactose it's not going to function at all. Okay, its function is going to be inhibited in the absence of lactose. So let me first introduce you with these terms which are our PIO, ZYNA, what these terms mean. So first of all, our P here is our promoter gene. Promoter gene is a site where our RNA polymerase is going to attach, and this DNA is going to this DNA will get transcribed. RNA polymerase is going to help in transcription, which is attached here is going to help in the transcription, and our RNA polymerase is actually going to act as an enzyme in the whole process. Okay, and here our I is actually an inhibitor, and this is going to synthesize an accessory protein. We can see here an accessory protein. This is our repressor. P again is our promoter gene. O is our operator gene, and operator gene is the one which is actually going to regulate the expression of the structural genes. Now you are wondering what are structural genes. Here our Z, Y and A, these three are our structural genes which are present in our operon. Here our Z is going to transcribe and translate in beta galactosidase, and so it will produce this enzyme. Okay, and our Y gene is going to help for the synthesizes of another enzyme which is known as our permease. Here you can see it is permease. Okay, and our A gene is going to transcribe and translate our transacetylase. Okay, so these are the enzymes which are going to be translated and which are going to be transcribed and translated by our structural genes. So let us see what happens in the absence of inducer. This means in the absence of lactose or in the presence of inducer. So, and this also means that in the presence of lactose. Here our lactose is inducer. So here in the so first of all let's take an example in which our lactose, so first of all let's take the example in which our lactose is going to be absent in the absence of inducer. So here if there is not if the lactose is not present outside the medium of the membrane of bacteria, if there is no lactose, then the structural genes, we don't need the structural genes because they are going to help in the breakdown of the lactose. If lactose is not present, then we don't need these because uh, these genes requires lots of energy. These genes require lots of energy for the transcription and translation of the enzymes. That's why uh, if there is no lactose, so we are not going to waste our energy in the production of these enzymes. Okay, so what will happen then, if there is no lactose present, then our I, which is our inhibitor, it's going to produce an accessory protein which is our repressor, and our repressor has a high affinity for operator gene, and it's going to bind with the operator gene, and when it binds with the operator gene, it restricts the promoter gene to slide over the structural genes, and hence it prevents the translation and transcription of these enzymes. So in this case our chain here stops at there. Okay, so in this case our lac operon operates in the absence of inducer, or we can say in the absence of lactose. Now let's come to what happens when there is the presence of inducer. Okay, so in the presence of inducer, what happens? In the presence of an inducer, whenever lactose is present outside the membrane of the bacteria, then the lactose is going to be diffused in the cell by the help of the permease. So, as we know, as our permease is generally our transcribed and translated by the gene Y. So, here a question arises that how our permease can be present earlier in this cell before the transcription and translation of the Y gene. So the answer to this question is that at any given time, the cell has some amount of permease which allows the entry of lactose into the cell. So after the diffusion of the lactose into the cell with the help of the permease, our lactose get converted into another form of the lactose, which is known as allolactose. Here, this is our allolactose form, and our repressor, which is our accessory protein. Repressor have a higher affinity for the allolactose as compared to the operator gene. So it attaches to our allolactose. So it attaches to our allolactose instead of our operator. In this case, our operator is free here now, and our repressor get inactivated. Now our promoter gene can easily slide over the structural genes, and our these three enzymes can easily be now transcribed and translated. In this case, this whole DNA is going to be transcribed. This whole DNA is now going to be transcribed. And at the end of this, our lactose will break down into glucose and galactose. Thank you. Have a nice day.